these
Pleurocorallium.
Yeah,
Steve
corallium.
Says. Pleuro.
Pleurocorallium is right. And
Thanks, Steve.
yeah, thanks, Steve.
Any further, or are you?
No, I think we got it. I'm pretty sure Steve's right on this. I couldn't quite come up
Roger.
with the name. Thanks.
So the polyps just retracted on that one?
Yeah, that one used to be Pleurocorallium kishinoye, because that's the
what
only
he's
white
just said,
one.
Yep.
Oh, you just said that?
Yep.
Okay, yeah, all right.
Wow, named after Senator Noye?
No, I don't think so. I think there's another Kishinoye, that's the researcher's name.
Ooh, we're getting lots of typing going on in the chat now.
Yeah.
Lots of input. It's exciting. So if you don't know, we have a very cool science chat that allows all sorts of researchers from around the world to participate live in our expeditions and provide us with expert commentary to augment our, or at least my very scientific terms such as zombie fish and things like that.
I'm going to make a point here, Steve. In the gallery, the name I think was changed to Pleurocorallium porcellanum. And I always knew it as Kishinoye as well, but somebody contacted the group in Hawaii and told them to update from Kishinoye to porcellanum.
That's what Steve said at the same time. Same time you were saying it.
Yeah, two minds thinking alike.
Uh-huh, yeah.
We're both double checking.
That's right, you get has to be correct then.
There he goes, yep. He was looking at his resources, I was looking at mine. You know? I was thinking the name had changed, I just couldn't remember.
And for those at home, you could look at the same kind of things we're looking at under the Okeanos Explorer Animal Guide. Just put that into Google.
How about, it looks like it's a stock sponge, isn't it?
Where are you pointed? I missed it. Oh, right the thing right there, yeah.
Yeah.
That's the chalice sponge.
I think this is a little bolosoma.
Okay, zoom in, please.
I think I see the osculum right now.
The osculum, that little hole at the
sponge
top.
off to the right, too.
Yeah.
Unbelievable.
Yeah, so this is bolosoma, and we've got specimens, of course, but it's nice to document it on the top of the seamount. Great, thank you. And the other guy there.
We've seen a bunch of those.
That's, I don't know, that's a curious little bugger. I'm pretty sure it's been collected before. I'd have to go look it up.
Is that a tiny little Corallia?
Yeah, it could be. Man, you are getting good.
Thanks, man. All right,
way to go.
Give him a cookie.
I'm sorry, but I have not even given any effort to learning the sponge names.
What?
I gotta pick my battles here.
How could you be with Hui Huakai then?
Because I know that you folks in the back have it totally covered. Full faith.
I'm okay with you sticking with corals. If you nail them, that's fine.
Where are we going? I'm sorry, I don't have any idea. That way. Okay, thanks.
Looks like a Paragorgia.
Paragorgia.
Kenneth is kicking in on that other eel-like fish we saw with the little white dot on the head. He says...
Aldrovandria phylacra. Yep.
He says it's on the head of the halosaur, probably the dark species Aldrovandria phylacra. He says it's a clear window through the transparent skull bones, allowing light to stimulate the pineal organ. Any one of those. That area is occluded by black melanophores in males during the breeding season. It begs the question of just how deep the last few solar photons penetrate in the ocean, maybe deeper than the theoretical 1,000 to 1,200 meters in the clearest ocean waters. Wow. That's fascinating.
Yeah, thanks, Ken. I totally agree, Aldrovandria phylacra. I just wasn't going to go there because it was so fast to look, but...
There's some very cool adaptations for all these deep sea animals.
170.
Oh, Chris, do you mind repeating the name of the fish that was dying down there, the silver fish?
Ken said it was Omosudis lowii, so Omosudis lowii.
Oh, there's another one of those
L-O-W-E-I.
corals. Is that
Okay.
different from the
Thank you.
other one we saw, Chris?
I think it's the same thing.
Same
I
one.
mean, Steve thought he saw one of these on the previous watch, and I'd agree. They're kind of weird little coralliids. They're not like your pretty fans that we've been seeing.
Why do they all have their polyps retracted? Is that
They're sleeping.
they're sleeping?
I have no idea. Just
Have a nap.
It's not lunchtime.
The zombie fish is also known as hammerjaw as well, or no?
what?
The
let's see.
zombie fish that we saw at the beginning, dying.
That's it. That's the
Okay.
one, the Almosutis loweli. That's the wait,
Hammerjaw.
hammerjaw. Yeah.
Hammer? John?
Hammer jaw.
Hammerjaw. Cool.
It's it's a good boxer name.
Hammerjaw.
Hammer jaw.
That is a good
Ooh,
boxer
what is
name.
that?
What?
Gone.
Dancing ring. That was really
Nice.
interesting. So what are these sponges that look like the Balisoma?
Yeah, they're in the family Pheronematidae, and I'm pretty sure I've actually looked at one that was collected that's in the Bishop Museum.
What was that black thing behind?
I
What was the black thing behind the sponge that we just looked at?
I didn't see it.
Is it possible to look?
I don't
Oh,
think
too late.
so.
No, it's fine.
We can if we really want.
It was like something moving on the ground.
Oh.
I missed it, I'm afraid, Aaron. Sorry.
Come on.
I know. I every time I look down at the iPad,
this
I
one.
miss something.
Let's look at this one.
Whoa,
Oh.
shark. Here we go.
Zoom in,
Oh,
please.
That's a shark?
nice.
And now that I can see it, I agree with Ken. This is Centrocilium nigrum. And I think that's what he thought the other shark was, or at least that's what I think it is. Let's see what he says.
Whoop.
The girth on it.
Wow, he
Yeah.
is not slow either.
Yeah, it's good.
Gone.
Gone.
They're probably attracted by that dying fish. It's no wonder there's a couple of sharks. It might be the same one, but it's very possible it's another one.
Yeah, I'll bet it's probably the same one. He's just interested in what the heck we're doing and what we are.
I think he's after that zombie fish. It's a good meal.
The two of us combined, that's a good place to be.
Mm-hmm.
Zombie fish and ROV.
Oh, what's that sponge? Is that this? Oh, that's the same one we saw earlier with the.
Zoom in, please.
Oh, what was that? What was that called again, Chris? The
Postolastra. It's a demisponge.
demisponge, Post-Alaska. Post.
All right, thank you.
Where the heck is this?
Demisponge.
Trying to find that shark in the guide to
Oh, I see.
show the
Nice
spelling on it.
looking rock here.
Another one of these. This is a Paragorgia, I think.
Dead or sleeping?
Oh, there's another
the
shark.
It's possible there's a bunch of sharks up here. It's kind of their turf, I think.
They're telling us that, that's for sure.
And they don't like you being an interloper.
No. Show of force. Get out of here.
You don't think it's more this one, Chris? This
Excuse me?
het mop.
Well, that's what I
Etmopterate.
suggested, but I was going with Ken's suggestion
Oh,
here. And I
okay.
can't even find Centrocilium in the guide right now.
Coming out to a
Oh,
little
there it
slope
is.
here. Seeing some
Yeah, the head
Argus
was really
sonar.
kind of
I
beefy
like
and
that guy, I
thick.
think.
sonar.
Argus is at 225.
Head up there, head up.
We can
there,
go up
there.
that a bit if you want.
Yeah.
All right, so 225. Is that what you're at, Argus?
Chris.
Sure. Why is it on the left? Does that make sense?
225 is on the left, so it's a little bit
More south?
like 190.
Yeah.
Back to sorting out
Or
the very similar
210.
squalid shark.
That's just, I mean,
Let's go the way you're going. What are you doing?
I'm doing
Here?
195.
195. All right,
Yeah.
let's do that.
Ken was,
What's this little yellow thing?
take a look.
Zoom in, please. I'm guessing zoanthids.
And you would be right. And what are they growing on? Oh, wait,
coral.
wait, I'm not so sure. We need a little bit closer zoom.
Okay, push in a little more, please.
Is Asako on? She may have signed off.
Steve's typing.
Yeah, Steve, give me a hand here. Are these zoanthids, or is this an acanthogorgia or
Plexaurid.
plexaurid?
Plexaurid,
Good.
All right,
sure.
come wide, please.
Thank you.
Coming up.
What are those guys? He was kind of telling me how to identify those plexaurids the other day. He was saying they have sort of a white base and right at the base of the polyp stalk. I got to look at that more closely.
What plexaurid? What genus are those?
The line and the rock coming up.
Is that the genus?
Steve, we've collected a bunch
That's
of plexaurids, and they're sitting around in museums, and nobody's working on them. That's why I'm a little reluctant, even if we got a new one.
What's that? Yeah,
Oh, there we go. Plexaurid.
but I could follow that for a sec. All right, let's go to here.
So you said it had a little white around the,
Yeah, something like that.
base of the polyps or the base of the...
Yeah. Maybe you would know if Asako made some comment on
Aaron, can
another
you zoom in
watch
a little
about
bit, please?
something that she thinks would be really good to collect.
Yeah, good there.
Sorry.
Ah, rats. Bonk.
Oh, let's zoom this, please,
Whoa.
the crinoid.
Sure, this big leafy thing? Go ahead,
Yeah,
zoom.
this is...
Right smack in the middle, yeah.
I thought I saw one of these earlier, and
Wow,
for the
that's
life of me, I
nice.
cannot remember the name. I've got to...
It's really dense,
Yeah.
dense arms.
Yeah, a lot of arms. Should we put ship moves in still, or do you want to possibly sample? Oops, sorry, come wide a sec.
I don't think we want to sample, but give me a second.
Okay, push in again. Thank you.
Where are you? Oh, it's a stock. Believe it or not, it's a stocked crinoid. That's why I couldn't find it. Okay, it's Indoxocrinus species, and it would be a good collection, I think.
Okay, come wide, please. Roger that. 1,000 meters will be all good.
I don't think I've ever seen one collected here, and it only goes to genus. So it's called...
That's beautiful.
These are really weird because they have sort of a modified stalk. I think they can almost walk around on it with these little tendrils coming off it. You'll see it when you collect it.
Is this a slurp action, or what's the scoop?
Well, they're not really attached. I mean, they kind of stand there, I think, if I'm not mistaken. Will
So-
that slurp okay, or is it going to all bust up?
That's the question for you.
How about trying to grab it and see what he does?
Okay. Ah!
Do these have arms in multiples of 5?
I don't know, but he's got a gazillion arms. If you want to try counting them on that picture, not a gazillion, maybe a billion.
1 billion.
Half a guacamole.
Coming for you.
Yep,
Coming for you.
it looks like it.
Oh, let's
I
see what he
counted 20.
does.
Stand
I
by.
just have no idea because we've never collected one of these before. So I don't know what he's going to do.
Okay,
I
zoom in
don't-
there, please.
I don't think he can swim, so. Yeah, Steve is also agreeing. He didn't think it would slurp well. So let's try it.
Do not think it would slurp well.
Yes, that's correct. That's
Roger.
what I was thinking too.
I don't even know if I can grab this guy. Hope he doesn't leave.
I don't think he swims. He walks.
I'm going to have to do this differently. Come wide, please. I got that rock all interfering. Let's try this way.
You can.
Just do it totally blind. That'll be good. Can you put bubble on that, please? What could go wrong? You can zoom in and darken that too, please. Yeah. And you can zoom in on Zeus too. Yeah, that's great there. Thanks. And then can you iris it down too, please? That's shift zoom. Shift's top left.
Did you get this, Kainalu?
Yes, I did. Thank you.
Again, those shifts engaged. It's got the light.
And which is it? It's under crinoid?
Yeah, nice.
It's stock. So you're going
Good
for
there, perfect.
feather star. It's actually a stock crinoid. So you have to go in the other group, the sea lilies.
Oh, okay. Oh, I see. All right.
Come on, man.
Stock
I want to chop your
crinoids.
arms off.
Right. Sea
chop your
lilies are
stock
stock
off.
crinoids.
Knock my block off.
Knock your block off.
Come on.
You got 'em.
Come on. Come on. Work with me. Yeah, there you go.
Nice.
Yeah.
Oh,
All right.
grab. Nice
So
grab.
you see he's not attached.
Stay zoomed in on Herc for a sec.
I wonder if you could twist the arm a little bit so we can take a look at
That's
him.
the game plan. Yep.
Yep, great. Thank you.
Nice.
Okay, zoom in more, please.
Yeah, this is great. That's a beautiful specimen.
Nice.
Well, I guess I'll put him in the starboard box, question mark?
Yeah, we have starboard A, B, or C open.
Okay, anything floaty in there that I should be aware of?
Nope, just rocks.
Rocks do not float. Okay, come wide, please.
Excuse
You can
me.
put bubble on the
What's in
starboard
those
side
boxes?
Just rocks, it looks like.
Is there any open box?
Yeah, there's three on the starboard.
Oh, okay.
Yeah, I
A,
was
B, and
asking
C
if
are
there was
empty,
floaty
right?
stuff.
Yeah.
Yeah, rocks will not
Rocks will not float.
mix
Tip
with
up on
this
them.
guy
Yeah, thank
well.
you.
Thank you.
Rocks will
And
get all smashy.
Some rocks float, pumice. Floats.
Yeah, okay. Yeah,
There's 3
technical.
open boxes.
Okay,
A,
sample,
B, and
please.
C are open.
Yep. Sample, please. Okay, go ahead and open. Okay.
Wow, terrific. That's going to look great on my mantle.
Oh,
wait, I guess we got to put it in the museum.
That's it. We're
You
going
can
to
give
lock
me
you
that,
up,
please.
Chris. Permit violation.
Hey, Data,
I'm
what's our
bringing you
sample
to
number?
NOAA jail.
That was sample 118.
guarantee the
118.
food's not as good.
Thank you.
And that was box A, correct?
Aaron, in video,
that was box A, correct.
Thank you.
we have a question for you, Aaron. Are the color stripes on the arm used to color correct the camera?
Okay, here we go.
Yeah, so we do a white balance and a black balance at the beginning of each dive, right when we get down there, and that's a good reference from when you're looking back at the video as well to seeing if your colors are the true colors.
One more time.
Thank you.
I don't suppose we could take a peek at that star, could we? I know you're backing away.
I'm easy.
I'm going to be a pain in the rear end today if there's this many things to look at. So I'll tell you, there is. These are all different, very different community than what we've seen.
Yeah, these are like pillow lavas again.
Yeah. Those are demosponges again.
Little shrimp perched on the lava rock.
Yeah.
Coral field, look at
Oh,
that.
yeah.
Oh,
Orange,
yeah.
too.
Oh,
We're all
coral.
exactly the
black,
same age.
Black
Oh,
coral.
wow, they're all over the place.
Cool.
Coral forest.
These are plexaurids, not plexaurids, primnoids, Calyptophora wiveli. Good mentioned at the last watch that these are kind of an unusual primnoid because they have what's called a biflabellate structure, which means if you look at them, if you collect one and you look at them, which we're not going to do because we have some, but they actually form two parallel fans, kind of like a hamburger buns or something. And then also when the polyps close up, they close down, where most Calyptrophora, the polyps close up. So it's kind of a very unusual guy. I think one of the reasons it's in that genus is because it has a certain number of scales along the stem that's characteristic of that genus. It has a smaller number of scales along the stalk than Norella or Candidella or something. Anyway, that's more information than anyone wanted. So sorry.
No, that's great.
Paragorgia.
Crinoid ladder.
Trevor, we have
To the left there's a sponge, if you could zoom
Left
in.
is
Just went off screen.
Left.
That guy. They had one close-up of it, but I just wanted to get another one. And the last watch they had one close-up. These guys are, believe it or not, these are a vase sponge.
Okay, zoom in, please.
Doesn't look like it, but
The little cornucopia looking thing?
yeah. Has a tiny little vase that just goes down to nothing with a tiny little stalk. And this is called Atlanticella, which is, perfectly logical here in the Pacific.
Boy. That's just, what am I doing? Who's driving this thing? Come on, please. I'm sorry.
Atlanticella.
Did you see what you wanted, or you want me to try again?
No, that's fine. I just wanted to document it again. So that's Atlanticella. And obviously it was presumably genus was first described in the Atlantic.
What does that sound?
Sounded like a dirt bike.
Yeah, motorbike driving by, I think.
That's a new one.
Yeah, that was new. What?
Where is it?
huh?
It's just a Harley driving by. This studio that we're actually on land for. Faking the whole thing, yeah.
Sorry. This really isn't deep sea exploration. We have a studio in our
Steve
basement.
thinks it might be a good time for a Niskin.
Niskin!
That's a good point. Thank you, Steve. Let's do
Niskin.
that.
Why don't we do a Niskin?
Why don't we do a Niskin?
I'm surprised we're not doing a Niskin right now.
In fact, that's a great idea because we haven't been in a field of these corals and they're all uniform.
Yeah.
That's really good.
So the Niskins, again, are water samples. We'll pull a little trigger, which opens up and allows us to collect a water sample from this area. And that allows us to analyze it through a technique called environmental DNA, or eDNA. It basically is we look at the water sample, see what kind of genetic sequences are in that water column, and you could potentially identify organisms.
I'm going to do 2.
Yeah,
from that.
Open.
And even organisms that are hiding in rocks and other places that we don't even see. So it gives can give a bit more comprehensive picture.
Before we go, if we could do a snap zoom on that white sponge on the boulder.
Yeah.
Thank you.
Right on that rock that looks like a crocodile.
Yeah.
Crocodile rock.
Crocodile rock.
Where'd you go, Herc?
Go ahead, zoom.
Yeah, I think, Andy, I'll let you handle this one.
Yeah?
Yeah.
I should know it?
No. We're not going to collect this because I have to look this up. I've forgotten what it is, but thank you. It's another thing that we haven't seen so far on the cruise, so it's good.
Ooh, that's exciting.
Okay.
New sponge.
Okay, what's that bearing? Okay. 160. That's super cool. Super cool. The reason I like Paragorgia is the color.
Mm-hmm.
I also like that purple one, which I can't remember the name of now.
Victor Gorgia.
Victor Gorgia, yes, thank you. $160, you say?
Yeah, I am not finding an easy match for that one, Chris.
Yeah. Oh, we got a fish.
Fish.
And probably a halosaur again.
Gone.
Closest section is the Rosella other
Well,
in terms of morphology.
yeah, it could be a small Seca calyx, this guy right here, which I think probably would.
Oh, is it stalked? I thought it was like just kind of...
No, when he passed by it, I could see it was on a stalk.
Oh, okay. Oh, I didn't catch that.
And it was a very small one. These guys can get huge.
There's another little fish.
Another one. Oh, they're all
another
swimming
fish.
downhill.
Fish.
That's a lot of holosaurs.
There you are.
We have some historical commentary. So for Niskins, Shale Niskin was the inventor of the Niskin bottle back in the early 1970s.
Name was Shale?
Shale. He married the daughter of Captain Sid Hartshorn, captain of the University of Miami research vessel Columbia Iceland. Still sailing in the 80s, Sid was one of the last to be licensed as a master of steam sail and motor vessel. Thanks, Ken.
Yeah, those are pretty cool names. Shale Niskin
That
and
is.
Sid.
That's the most sciencey name ever.
Yeah.
Shale Niskin. Sounds like they should be an NPR reporter. They always have the most unique names.
Totally.
Well, this is kind of developing into a little community
Nice.
here, isn't it?
Leaf sponge. What is that? What is that one almost in the middle right now? Is that, oh, it's two.
These are those Poeciloster, I think, these ribbon sponges.
Oh, ribbon sponge.
What's that kind of pasta that does the twirl again?
That's exciting.
Rotini or something?
Hmm?
That's what this is.
Mm.
Yeah. If
You got
really
dinner
should-
on your mind, Trevor? Rotini sponge?
It's not like we're going to eat in 6 minutes from now or anything.
Why? So what's the benefit of these attaching low and wide, Chris, for these sponges up here? A lot more current near the top, or what?
I don't know. I don't think there's particularly more current. There may be less current, and they're trying to spread out a bit. These things don't, they don't have stalks, these demo sponges.
Well, it seems very stable configuration.
Yeah, it is. And sometimes they'll form lines, as you can sort of see that. Oh, there is a stalk guy over here. I wonder if you could take a look at him. Oops.
Want to zoom on it?
Sure, let's zoom on him. Probably a Calophacus, or a Bolosoma, one of those two.
Okay, zoom in, please.
Oh, what?
Pretty far, but see what I can do.
Bolosoma, that's good. How about the thing up to the right, that
Yeah,
yellow
why is
thing
it
above
moving
it?
so much?
Little yellow thing?
Yeah, what is that?
It's the
There
jiggy
we go.
jiggy.
So there is
Jiggy,
some
jiggy
current
growl.
here, obviously. Look
yeah.
at that guy going.
I know.
All right, Steve, are we talking plexaurid? Are we talking acanthogorgid? I don't know.
All right, come wide, please. Trim.
Yeah,
So a viewer wants to know what's the difference between the Van Dorn and a Niskin. Van Dorn and Niskin, what's the difference?
the hartshorn.
Isn't it the same
well,
thing?
the bottle was named after Shale Niskin, I guess, but he married the daughter of Captain Sid Hartshorn, so I don't think there's anything named after Hartshorn. I don't know. One of these big Pheronematids. But it's a different one. See, it's more solid again, not quite as holy. But if you come around into the cup, you'll see all those typical holes, I think.
So this is the same that we saw on the other dive, except these are all tilted over because of the current. The current's much stronger.
Yeah, but also this could be a different species actually in that group, and I think they are.
They'll have that laid back,
There's a good current up here.
spongy lazy boy kind of structure.
Trevor, what's the current like? Is it a strong current?
It's strong, yeah.
Okay.
Mm.
Is it coming from the east or west?
Coming from where I'm facing now. What's that? Coming out of 225, so it's a 045 current.
0.5, okay. So sort of northeast.
Yeah. Yeah, I'm driving straight into it now. Maybe a knot?
Let's see, Herc heading, okay, C2.
Bridge, I have another 20 meters, 160.
Northeast, I guess.
A viewer wants to know if there's a chance to see an octopus in this region.
That's actually a really good question.
Dark spot.
Because we're seeing a lot of fish.
Yeah.
Ooh,
Dark spot.
dark spot.
Maybe it's
Rock?
octopus. No.
Could be a rock if we're lucky. Zoom in on that rock, please.
A lot of the fish are sort of low oxygen specialists here, and I'm not sure the octopus could tolerate the fairly low oxygen here.
Oh,
What is the oxygen here?
thank you.
I'm trying to see. Let's see. Concentration.
Temp, probe, oxygen. 44? Is that
Yeah,
it?
micromolar. See,
Micro.
you multiply it times 32 and divide by 1,000. You got a cell phone with a calculator.
Sure, 44 times 32 times 1,000.
Yeah. So you basically
not
multiply the molecular weight of oxygen, which is 32, and divide by 1,000, because what we're seeing on the screen is expressed in micromolars, and that's how you can.
1.408.
Yeah, so it's quite low, very low.
Hmm.
It's only going to get down to about 0.98 or so, probably here. So
Yeah, it dropped down pretty significantly from the beginning of this dive. It was up at 44.5, and now it's diminished about
Can I reset?
1.5.
Yeah.
0.5.
I think USBL's simmer down a little bit.
Oxygen saturation's dropped off.
Give it a stern talking to.
Settle down.
Settle down, you. All right.
it almost looks sort of volcanic, doesn't it? I don't know. It's sort of strange.
Let's go look this way. This is completely the wrong way.
There is no wrong way.
That's true.
Yeah, but there's a cool sponge. How about this guy over there?
You want me to look at it?
I'm trying to determine whether it really is cool or not. It may not be cool. Yeah, let's
zoom
take
on the
a look
maybe
at
cool thing, please. Well, it's got a hole in it, so it's in its punk phase.
Oh, yeah, it's just one of these demo sponges. Yeah, somebody punched a hole in it. Well, I have no idea why the hole's there, so it's kind of...
Maybe it's a holy sponge. Demisponges.
So just to give the viewers a heads up what we're trying to do here, we're sort of running along the eastern edge of this flat top seamount, which is a guyot. It's called Tamana Guyot or Tamana Seamount. And we're headed to what we think is a very small volcanic cone structure that we're going to get to. It's about 1 kilometer away from when we started. And so we're just running along this edge. It's hard to see much of an edge because it's sort of gradual. But then we should see what's living on the cone, which is a little higher topography, and we might have some other types of animals that want to get up a little higher living on that cone.
Urchin.
Where's the urchin?
Below the lasers.
Oh, yeah. Kind of third. This
Zoom in
is
on the urchin, please.
this is Sparosoma.
So what are they, if they're not scraping algae, what are these urchins eating down here? Oh, there's another one beneath it. Look, there's 2 urchins.
What?
2 different kinds.
Oh, yeah. So that's,
Wow.
I think that's, I was trying to remember. This is Aspodiadema, I think, or something like that.
Yeah, it looks
I
like a
gotta look that
diadema.
up.
Okay, come wide, please.
Thank you.
Thank you.
Let's see. Oh, here we are. Yeah, Aspodiadema, I think.
Where have I seen you before?
There you are.
Nice.
Oh, another, yeah, there we are. Another Endoxocrinus.
Got it.
So what would the urchins eat down here?
Oh, there's got to be something that's on the rocks.
Are they just
Yeah,
digesting material on the rocks, or?
Uh-huh. Yeah, I would assume. The sea star, I don't know if we can stop for that, but
Very. Oh yeah, absolutely. Go ahead and zoom in, please. It's pretty far away.
boy, I got to do a little looking up on this thing.
You need a better imagery? I can get a better.
Yeah, maybe.
Okay, come wide, please.
Aspodiad. Do you know, Steve, what they eat? That aspodiademid?
Do I know what to eat?
Oh, I was asking Steve because he was posting about the sponge.
Okay.
Might have gone to dinner. Hey, you can see the current still howling. Look at that crinoid up at the top view.
Yeah, totally. Okay, zoom in, please.
What are you?
Ah, what am I doing? Who's driving this thing?
I think it's something called Tarsostrocles, but I can't seem to remember where that guy belongs, what group he's in. Let's see. Oh, here we are, I think. Nope, that's not.
What is happening? This is not going to work. All right,
Current. It's
I'm out
kicking
of
you
there.
around.
Bye.
Okay, thank you. Where are you? I don't think he's that.
We're having a little bit of rotation here as some of us get spelled for dinner.
Which way am I going, eh?
Yeah.
160, roger.
Um,
160. Which way am I headed now? Not 160.
Yeah, I'm afraid we're gonna have to stay with Sea Star on that guy.
Sea Star.
Yeah,
Okay.
sorry.
No, no worries. Thank you.
Roger.
Okay, so we're going slow here because we're poking around and looking at stuff if we could, Dan.
The boat?
Yeah, we don't need to make any distance. We got plenty of time today if that makes any
Okay.
difference. And we want to get down close to the substrate where everything is.
Roger, sorry.
Thank you.
Okay, I'm getting pulled.
I'm, where am I? Am I really way out there? Why am I going that way?
Just approximately 100 miles away.
Totally
Give or
100
take.
miles away.
I'm just going to wait till we get sorted.
So the boat's stopped, correct?
It finished a move, yeah, it's just hanging out.
Give me a second to
No worries.
find the seabed. I think it's here somewhere.
Oh, that was a nice shot of topography. Okay, cool.
For some reason, we're way off to the,
Way off in the rhubarb?
yeah. Can you kill your auto head for a second? I need,
Sure
or
thing.
come down or something. Yeah, come down.
Yeah, I definitely will. I'm going to actually keep auto head on when I come down.
Yeah, sure. Just need it.
I just don't want to go flying off around in circles.
Some slack, man. I can't
I'm cutting you some slack. I am uphill of you, so my altitude's 13 meters now.
All right. Which way do I need to go here?
Backwards. Backwards uphill. Should be no problem.
How'd that happen?
Or you could just turn around. That's cool. Go that way.
I can't turn around right now. I don't have, Argus is still swinging to the south there. Looks like the boat is moving.
If it is, it's jiggling just the littlest bit around a position.
Yeah. So we're headed to a little cone feature that looks like it's a secondary cone, but we're taking the scenic route right along the edge. In the
okay,
eastern
I'm starting
edge
to see that you
Current's
have maybe a
coming
little
from
bit
the
of
northeast.
Give me that
according
other,
to
I want that other 3 meters.
Trevor.
Yep.
I still can't get there.
You can have it.
I'm trying, it's not happening. I'll just keep
There
flying
you go.
backwards. You're gonna have to spin around. For me to spin around,
Yep.
I can't do it.
Where's tail to tail?
What's that?
We're tail to tail.
Are we?
Yep. And I've got auto head on, just being on the opposite heading.
Come on, Herc, you can do it. You can do it. No, we can't do it.
Do you want me to move the boat closer to you?
Yeah, sure.
Would that make things better? All right, how about 20 meters, 130 towards you?
Yeah.
Bridge nav, can we move 20 meters, 130?
I can see Argus's glow.
Yeah, I know.
behind.
I actually saw Argus in Herc butt.
Sorry guys, the problem and girls that we're stretched out tail to tail here and still swinging south, so I can't seem to get enough slack to spin around and look uphill.
Okay.
And Herc doesn't go backwards very fast. It goes forwards good, but okay, try that.
Nice. Okay.
Finally. So it was Aaron moving the boat that saved the day there.
Yep.
Good job, Aaron.
Thanks.
The substrate is always kind of curious to me, whether that's just all manganese crust. I don't think there's lava or anything under it. Could just be carbonate or just sort of draping over the, almost like draping over the sediment.
Yeah.
Okay, Gabby, headed towards you.
Okay,
Yeah.
I'll start to bring my head around to 160.
Yeah, and again, let's go slow and as low as you can because we're looking at lots of stuff.
Slow and low.
Yeah, thank you, Dan. Appreciate it.
I liked your shark.
Yeah, it's pretty cool, wasn't he?
Yeah.
And
Good shape. Pretty good shape as opposed to that zombie fish.
yeah,
Look at all the little shrimps.
Why don't you can just turn around
that
and
shark
look at
is
Herc,
always
Gabby.
in the OMC.
Okay, you don't care
Every
whether
time
I'm
we've seen him.
Nope.
looking 160 or not?
No, or
Okay.
I'll stop. Might as well find Herc. Do it tether-friendly fashion if you can.
I'm still looking
Sure
for
thing.
things that we haven't imaged already. So I'll keep an eye out for something. And I was watching some of your previous video too, saw some really interesting things as well before I came up.
I'm holding on moose until we're well sorted, yeah?
Yeah, it'll be a while if they want to go slow. So, where are you going? South?
Yeah, we were heading kind of southeast. It just finished that move that I put in to get an overview, but now we can go
Oh yeah, if you want to take that back, you can.
about 145.
Sure.
Cool. Bridge nav, can we get a 20 meter step 145?
One of the little Alanacella sponges that we've seen. They imaged it
20
on their
meters.
watch, and we did too.
All
They're
right.
still hanging around. I think when Beth comes on a little bit later, she's going to have plenty of material for her crust stuff. Then they look, I think it's very altered.
Yeah, we collected one for her, so she's got 2 in the box now.
Oh, is that right?
Yeah.
Oh, okay. She should be all set.
Yeah.
Another one of these beautiful Calyptrophora wyveli that we just talked about a few minutes ago.
Or do you like a close-up of that guy, or keep going?
I think we're good. It's a we got a close-up of it so I could sort of explain it a little bit.
Sure, go ahead.
Yeah.
I think he said that he already did.
To the right of that, let me circle it. There's a little star. I think he's right there. Oh, no, sorry. Wrong one. Here. That would be a good close-up.
Okay.
How'd you even see that?
I don't know what he's talking about. So.
It's a little white
Gonna go
spot
where
on
he
that rock.
circled
Let me circle it. Where'd he go? Right there.
I think
Oh,
that
almost.
was
Okay.
A little tilt, yeah.
And
sediment.
Sediment.
oh my goodness, who said that was a starfish? Jeez. Emil, come on, Emil.
I come in here and just cause all kind of trouble.
Yeah, sorry about that.
You
There
did-
is a little star down there. Now, trust me, there's a little star down there that kind of looks like that, and I saw it on a previous watch, and I thought that might be one of them.
I believe.
So no negative on the close-up here. Keep going.
Yeah, that's fine. Thank you.
Works for me.
I get fooled by sediment in the Argus cam. When you're looking ahead, see what's coming, you think you've got this giant sponge and it's
Sediment pocket.
sediment pocket.
Well, this
Yeah,
one,
that got me a few times tonight.
it almost looks like there's a string of sponges in the Argus cam right now, and there's sediment pockets. It's just very deceptive substrate.
145, you said, right?
Oh.
Yep.
This guy right in front, it's an unusual posture. We collected one of these, but it's got a good view of the feed if we could zoom in on that.
Yeah, can do.
And come around the back side if it's possible to do that. We got the front side like that, but if we want to look at the rear end of them.
That was an amazing collection
Yeah.
that Trevor pulled off there.
Thanks, Aaron.
And after that, we'll get this guy first, and then that one to the right of him would be a good zoom too.
Can do.
I just want to show you these rather unusual stock. Believe it or not, this is a stocked crinoid. Who would have guessed?
I wouldn't
We were
have.
seeing a bunch of the bright orange stalked crinoids earlier.
And this is different.
This is different.
Yeah.
Is that the little one in bubble camera?
That one
Where's the
right
little one?
there.
There he is. I don't want to land on the little one.
Now look at his stock. It lays flat against the substrate, and it's got these, I think they're also called Syrian crinoids.
Zoom in there.
And I think he can walk.
What?
You can walk?
I think so. See that? So that's his stock right there.
Whoa.
And then he's got these little Siri type feet coming off of the edge. I don't remember what the name of them is.
Push it
There's his stock right there.
Bridge nav, are you holding position?
Yeah.
But it just walks along the seafloor and just
Well,
finds a new place to hang out?
I think he can walk, yeah, or I don't know, but he's like a hybrid between a stock crinoid and a feather star. And they're so weird, that's why.
That's crazy.
Yeah, it's crazy. That's a beautiful shot, Dan. It showed exactly what I wanted to show.
Roger that.
Let's back up a little for the other guy.
Yeah, this one here. It may be another one of these, but let's take a look at it too. I think it is a smaller
Yeah,
one.
it looks like we're moving off to the west.
Roger. Okay. Zoom in there.
Has anyone ever gotten on camera them moving
Closer.
along the seafloor?
I don't think so, but
Okay, I'm coming down. I don't have a ton of room to come down though.
but I'll bet they do.
Yeah,
You know, the feather stars can walk around on their corals, and they can also swim. And so I'm pretty sure this guy can
Yeah,
walk.
it looks like the ship might be moving to the northwest.
You can do a push in there.
It's what a nice shot, Dan. That's beautiful. Oh, look at his rock tilted too.
Yeah.
You see that? He
Yeah, I
rocked.
did
Wow.
see.
He's on a rocking chair.
Sorry, I'll stop moving the camera.
Easy chair.
That's so cool.
The ship's going on a bit of a holiday there, so we might have to participate.
No, this is great. Thank you very much.
It looks like we're holding now, but I'm not really sure.
Gabby so wants to pick up that rock that he's on, see if he jumps off.
The direction I think that we're gonna move
there's
if
that
we
eel.
follow the ship is actually gonna be pretty favorable to where Herc is right now. So
Bonus.
we can just keep an eye on it.
It's a halosaur, probably Aldrovandia.
There's another one or something swimming on Argus cam. You can see it.
Where is it
I need to
exactly?
come south, right?
It's like moving.
Oh yeah, I see him.
Just below.
Yeah, he's going to come into view of it. There he is. It's a shark. It's our friendly shark
But
who's been following us
Dan,
around.
expect Argus to move back northwest.
Roger.
So you don't have to move too far, Shell.
Go fish herding for a minute.
Oh yeah, fun.
It's I don't know what the common name for him. It's Centricillium nigrum. How's that? That'll help just about everybody, right? Yeah.
It's way too fast for me.
Okay. Thank you, Andrea. So I'm going to go grab some food, and Andrea's going to spot me.
Here's a shot of the stalked crinoid that crawls.
That's
Oh,
crazy.
it crawls.
Yeah.
Wow.
Same group.
Looks like I'm gonna come right under it, or kind of sort of under you.
All right, shall we try again?
Sure.
All right, I'm gonna still try 145.
Let me get out in front first.
Okay.
We're moving so slow. I'll never be able to catch up. Gabby's got to spin around too, so.
Roger.
Currently spinning.
It's some current blowing across here.
Yeah, that's what we've heard, and saw some corals like full tilt.
Yeah, if I hold my heading and let go of the stick, that's the current sweeping Hercules right there.
That's pretty serious.
Yeah.
Wow.
That's the current sweeping Hercules at
Half a knot or?
It's pretty strong.
0.3. 0.15 meters a second for those.
How many
metric
turtles?
people out there. That's got to be 5.2 turtles somewhere in there.
Okay, my head's pretty close.
Roger, I'm just moseying.
Oh, there's a shrimp and poop. Shrimp.
Scared him.
Moseying southish here. See if I can put a little in the bank against the breeze.
Okay, standing by.
Slowing down, I'm going too fast.
Got some good rocks coming up.
So what direction are we trying to step?
Nothing yet. We are waiting for Dan to get back in front of Argus, and then we're going to do 145.
145.
I can't believe we haven't hit the carbonate platform. That was a big old sponge.
I think we might be on it.
We just can't tell.
Yeah, I mean, it looks, Chris is thinking it's just kind of draped over the sediment. This crust. Looks like some more nodules down there.
Mm-hmm.
Did we stop going up and start going along a contour?
That's what we are doing. Yep, that was what Chris asked us to do.
Yeah, we're taking a scenic route to a little crater.
Cool.
Yeah, are we still going low and slow, or do you want to change that up?
Low and slow.
Low and slow, roger. Am I out far enough now yet, you think?
I was waiting for the middle, but if you feel good, we can go.
Waiting for the noodle?
The middle, like when you get in the middle of the screen.
Roger. Looks like I'm doing about 0.2 there. Somewhere between 0 and 0.2.
Flying.
Keep touching the rocks there, a little less Z bias going on.
All right, ready, go?
Sure.
Bridge nav, can we get a 20 meter step 145?
You want to see the curly Q one?
Yeah, let's look at the Iridogorgia.
They're always pretty.
Alrighty.
Oh.
That was quick.
It was quick. Okay, I'm going to hand you back over to the A-team here.
Mm-hmm. Okay. What? As what? You mean why is if it's basalt, why is it like that? Well, those are probably manganese nodules or manganese crust growing on top of a rock, but there's a hypothesis that we're already in the limestone cap rock. We're already in the wave cut platform. I don't know if that's true, though. The times I've seen come up on a wave cut platform before, that's very obvious. But that might have been a younger seamount.
Okay. What am I doing? This thing. Cool. Nope. All's right with the world. Sure.
Still got some
14321 or
current.
something I heard.